Hello?
Yeah, hi, Dr. Boles. I can hear
Hello?
you.
Can you hear
Hi.
me?
Hello, everyone, and welcome to Autism Specialty Care Education Series, which has been brought to you by India Autism Center and NeurAbilities Healthcare. Through this webinar series, we hope to cover a wide range of topics and provide comprehensive evidence-based information to assist educators and families in developing effective educational programming for their children and students with ASD. I'm Sakhi Singhi, Project Coordinator at India Autism Center, a first of its kind integrative community project coming up in Bengal. You can know more about it by clicking on the links in the chat box or following our social media pages. I'll also be attaching India Autism Center's brochure for your perusal. We also have here with us today Sandra Bean Lewis, Director of Marketing and Community Affairs at NeurAbilities Healthcare, which brings together the experts needed to provide evaluation, diagnosis, and treatment for individuals across the lifespan with neurological, behavioral, developmental, and cognitive concerns. This webinar series highlights many of the specialty care services of NeurAbilities and the experts who work together to bring patients of all ages personalized care. Dr. Richard Boles is a medical geneticist and a pediatrician who specializes in mitochondrial medicine, functional disease, and autism spectrum disorders. His expertise stems from decades of both clinical work and research at major academic centers, as well as from his most recent experience in cutting edge biotechnology and genomics. He uses an innovative and integrative approach in both diagnosis and treatment to best serve his patients. Dr. Boles will help develop the innovative Neurogenomics program at NeurAbilities, which is currently available for physicians and other healthcare providers, which Sandra will tell you about towards the end. Physicians and other healthcare providers will be able to consult with Dr. Boles directly about reanalyzing previously done genetic testing results, advice for managing neurometabolic disorders, or to obtain guidance on choosing and interpreting genetic testing. Dr. Boles served 5 years as the medical director of Cortigen Life Sciences and participated in multiple organizations, past and present, including the Scientific Advisory Board of United Mitochondrial Disease Foundation and a principal advisor of the Cyclic Vomiting Syndrome Association. Dr. Boles will be talking on the benefits of genetic testing for treating autism. We'll begin the webinar in just a moment, but know that we'll be fielding questions today at the end of the session. However, you can address your questions anytime by clicking on the chat box below. When you click on the chat box below, make sure you address your questions to the moderator, and we hope to have your questions answered towards the end of the session. If you have any general questions about IAC or NeurAbilities, you can address them in the chat box below as well. And Sandra and I stay back a couple of minutes after the talk is over to answer your questions live. There's an e-certificate of attendance for this webinar, and we'll be sharing the Google link right before the question and answer session. It is mandatory that you fill the link within 24 hours. Questions about e-certificates are taken on email or WhatsApp, and the contact information will be shared soon. According to our latest webinar policies, participant video will be permanently stopped if it appears to be unclear or distracting, and we'll be locking the meeting room in the next 10 minutes. Thank you all for joining us today. Dr. Boles, I request you to please share your screen.
Did that work?
Thank you.
Hi, I'm Dr. Boles, and I'm going to just start into this. The topic is on genetic testing and how that can help in people with autism. I'm a pediatrician, so often I'm going to say child, but sometimes your children are grown up, and so this applies equally to adults with autism. It also applies equally to people that are not necessarily exactly on the autistic spectrum, but have other developmental disorders, because it's really many of the same genes. And then this title slide shows is that we're trying to take genetic information here and then translate that to help patients with autism. And then this is, I just want to acknowledge and to thank the India Autism Center for this opportunity. Carter presented at 1 year of age following a vaccination. He stopped developing for the next year. He was later diagnosed with autism. His behavioral problems were, he had biochemical laboratory abnormalities suggestive of a mitochondrial disorder. Actually, most patients with autism have biochemical abnormalities suggestive of that there's mitochondrial dysfunction, so this is pretty typical. And then DNA sequencing revealed, there's tens of thousands of variants that are seen on sequencing, but it revealed one particular variant that I've been working on, which is in the TRAP1 gene. TRAP1 is a chaperone. It takes things where they're supposed to go and they don't do things that they're not supposed to do on the way there. Chaperone in the mitochondria that protects proteins in the mitochondria against oxidative stress. And I had noticed that this exact variant was seen in a lot of patients with chronic pain, fatigue, and dysmotility of the GI system. And autism was in many of the families based upon work that was done at Georgia Tech that looked at in 3 dimensions based upon the X-ray crystallography data of what actually the TRAP1 protein looks like. It predicted that a drug called Gancitron, which is also known as Kytrol in many countries, would help by turning off the mutant or the abnormal TRAP1 molecule, but leaving the normal one on. And then following giving Kytrol and Carter, anecdotally, he did much better. You can see in the green in the slide, he was more talkative, clever, focused, less aggressive, less sensory integration issues. He got along with his brother more and more tolerant of difficult situations. Before this, they'd never gone out to a restaurant with him because he would just run around and scream. But on Kytrol, he was actually able to sit there and to eat and not cause any trouble. There are 5 different times I know that he went off of Kytrol. And because either the insurance company didn't pay for it, or there was a nationwide shortage, or they forgot it on a vacation. And each time that he deteriorated in his behavior, and every time he went back on Kytrol, he got better. It was also dose dependent in that the higher doses did better. So he's been on Kytrol now for, I believe, over 5 years and still doing well on this. So Payam here was diagnosed at 18 months, also following a vaccination. It just happens to be that both of these cases followed a vaccination. I'm a moderate on vaccines. That means that I understand that vaccinations can sometimes be a trigger for autistic problems, but I also believe in vaccination in my own 4 biological children that have been vaccinated. Their autism can have many different triggers, including infections, surgeries, and occasionally vaccination. So he has mental health issues and functional issues such as constipation and fatigue. But like the other case, he was relatively uncomplicated autism, if ever such a thing exists. DNA sequencing revealed a variant in the creatine transporter. It revealed variants in many other genes as well, but this one seemed to fit. The stars I added there, these are where the family said that these are the type of problems that he had. This box down below came actually from the actual genetic testing results. And these are the results that you can see with the creatine transporter abnormality, which prevents creatine from getting from the blood into the brain. Creatine is sort of like a battery for brain cells. And particularly the autism and aggressive behavior fit very well. If you can't get creatine into the brain, you might be able to get cyclocreatine in there, because cyclocreatine can get into the brain without the transporter, where it's converted to creatine in the brain. And on cyclocreatine, he improved dramatically, and the green, again, are areas in which he improved in. And these were based mostly on OT and his teachers. And so that it's 2 different cases in which we're able to find an exact cause of the autism, or at least one of the causes of the autism, and we were able to treat it based upon understanding how that cause affected the individual. Now, who am I? I wear many different hats. I think of myself predominantly as a physician, as a doctor. That's what I've been doing mostly the last 30 years. I was 20 years in academia at Children's Hospital in Los Angeles and at USC, University of Southern California, as a professor. While I was there, I was a researcher, and I still do write papers and research in areas such as autism and cyclic vomiting, my other area of expertise. I am also the Chief Medical and Scientific Officer of NeuroNeeds that makes treatments, dietary treatments, based upon the findings that I generally see in genetic testing on patients. And I have been the medical director of DNA testing companies, but right now I just order testing from different companies for my own patients, and an expert in witness and legal cases. But predominantly today, I come to you as the director of the NeurAbilities Neurogenomics Center, where we are taking the latest in genetics to understand what's wrong with our patients and to apply that into therapy. A few caveats here. In the United States, we have the FDA. You probably have something similar in India that says, you know, if a drug is approved for a certain purpose, physicians are allowed, as long as they have a good reason and it's documented and there's informed consent, to use drugs that are approved for one purpose for another purpose. And certainly the use of Chitril was not approved by the FDA. It was based upon a genetic finding in a single patient. In personalized medicine in general, you figure out what's wrong with the genetics of the individual and you treat based upon that genetic finding. It's the latest in discovery, and there's no way that it's through the FDA. In fact, most of these cases is the first time or one of the few times that a drug has been used for that purpose before. Also, I'm going to be giving you my opinions on genetic testing and autism and other neurodevelopmental disorders. There are some official organizations that have weighed in and have written papers on what they think is appropriate testing, but those papers are now over 5 years old. They're in the dark ages and really don't apply whatsoever. They didn't even anticipate the type of studies that we now do. And of course, that the CNNH, now known as NeurAbilities, is providing genetic testing, interpretation, and treatment services. So I'm talking about DNA variants a lot here. Variant is just any change from the reference sequence. So reference sequences is what the computer has as the usual sequence in that position. And so theoretically, a variant could be 49% of the population, but it couldn't be 51% because that would be the normal reference sequence. Well, some of the reference sequence isn't exactly the most common sequence, but it was a sequence that is based upon the first few that were sequenced. And so occasionally a variant is more common than 50%. But in general, the variant is the deviation from the typical or the most common human sequence. One of the things when we deal with India is that the number of sequences of people in India is not as high as it should be based upon your population. And the reference sequence is too often based upon Europe and East Asia, where the vast majority of the sequences that made the database years ago came from. So people from India will have more variants simply because their genetics was not considered as much in the initial. That's changing, and in the last few years, we do have a India-based reference sequence that helps a lot. Again, unfortunately, it is not completely representative of all the populations that you have there. It tends to be more northern India in terms of the type of sequences, but at least it's far better than relying upon sequences from Europe or China. Variants can be small or large, and they can be rare or common. And so this is important to understand in terms of how we test it and how we interpret. So one base pair, I mean, DNA is about 3 billion base pairs from the mother, 3 billion base pairs from the father. So a single base pair is the most common variant of all. And it's the smallest variant that you can have. On the other hand, you can have millions of base pairs, like in Down syndrome, you have millions of base pairs which are extra, the entire chromosome 21. Somewhere around 50 to 500 base pairs is the difference between small and large. And the reason for that is because sequencing generally finds the small, and microarray, which at least in the United States was called CMA, chromosomal microarray, generally gets the large. Now, if you notice, there's this gap in here that neither of them found. So until fairly recently, we sequenced for small variants and we did microarray for large variants. And there is this gap where you won't find things. This is not very common, but there are mutations that matter in that gap. Today, I use whole genome sequencing for most of my patients. Whole genome sequencing finds variants from 1 to millions and everything in between. There is no gap, and it's a single technique that finds everything. And I do recommend whole genome sequencing, though it is not that, it's becoming more and more frequent, and you can get it in many different countries of the world. I know that there are patients in India that have had whole genome. I usually sequence from a lab that is in Israel, and another lab that's in Germany for sequences from the Eastern Hemisphere, but there are many other labs as well. And then it's just to show that, like some of the cases I've shown you, it can seem magical in what we can do these days. And what I do in reading a whole genome sequence was science fiction 20 years ago, and 10 years ago, I hoped that sometime during my career I would have this ability. My daughter loves to have her picture taken. But this is not magic. This is the machine that actually does it. This is an Illumina HiSeq 4000. The Human Genome Project that used laboratories throughout the world, finally in around 2003, was able to sequence the genome of the first person. It took billions of dollars and 10 years at least to sequence this one person. This machine can sequence a person in an hour. The machine cost $1 million, so sequencing is not very cheap, but it's coming down dramatically. To give you an idea of the scale, this right-hand side here, it's the size of a general refrigerator. It's about double the size of a refrigerator, this machine. So today I'm talking about neurodevelopmental disorders. That includes autism, intellectual disabilities, epilepsy, loss of milestones. It also includes many other things like ADHD. And so autism is the title on this, and perhaps why many of you in the audience are there. But some of you may say, well, some people say my kid doesn't really meet the criteria, or I don't think my kid really has autism. This applies equally. This is really looking at neurodevelopmental disorders in general. The reason I say that, and I'm going to show a couple slides of science here, is that the different neurodevelopmental conditions really have, to a large degree, the same genes that are involved. So the genes that are involved in epilepsy and intellectual disability and autism overlap with those of many other conditions, including schizophrenia, migraine, and many other conditions. In particular, the genes that are involved in autism are the genes needed to build and maintain a brain. And there are hundreds of known genes that when a mutation occurs or a disease-associated sequence variant occurs, that one of the results will be autism. And these exact genes are also that in other people can cause other neurodevelopmental disorders, and even disorders that are outside of the brain in many cases. And so what we're really talking about is we're talking about the genetics of brain disorders in general. This is another heavy science slide looking at a large number of patients with autism and the type of pathways that are involved. And I know this is a very complex slide, but there's two things I want to show from this. First of all is that not all patients with autism have the same genetics. In fact, they have many different reasons why that patient's brain has problems, why they have autism. The second thing is that all of the blue arrows are mine. All the blue arrows are on areas that are at least somewhat treatable. So many of the causes of autism are in pathways which we can actually do something about. So finding the exact problem in a child or an adult with autism is not just to find the problem and give a name to it and then put it away. It's to find the problem and hopefully find the solution. You can see that the two cases that I went through so far show that at least in some cases you can make a big difference. Okay, so another way to look at variants is rare versus common. A very rare variant was never seen before. And a very common variant is, let's say it'd be like 50% of the population. But what I'm getting to on this slide right now is I'm looking at the large variants. If you look over on the right-hand side of the screen here, you're looking at the largest variants. The largest variants are the ones that are picked up traditionally by chromosomal microarray. They could even be picked up by general chromosomal studies because they're thousands of base pairs, maybe even millions of base pairs in size. They're called copy number variants because there's either an extra copy or extra copies, or that you're missing copy or copies. These are the chromosomal locations that are, this was a study that was done in about 2016 or so, and these are the number of papers in the literature that are related to autism being part of these chromosomal abnormalities. And about 2016, I stopped collecting the data. There are many hundreds of different chromosomal areas that if you have extra or missing, that you can show signs of autism. Again, these are hundreds to millions of nucleotides. They can be de novo, meaning new, that they are not in either parent, or they can be inherited from one of the parent, in which case they're risk factors. They're picked up by microarray, but I've also shown they're picked up by whole genome sequencing. A very large copy number variant tends to have these problems. These are the kind of the hallmarks of what used to be called a chromosomal abnormality: intellectual disability or mental retardation, other neurological problems, seizures, hypotonia, include autism, growth abnormality, usually too small, but sometimes too large, birth defects of all different types, and dysmorphic features is when the face looks different, syndromic-like, just not the same as the parents, unless one of the parent is affected. This is one of the typical ones. On chromosome 22, the Q arm, the larger arm, at band 11.21. This is a common area. Patients that have a deletion or a missing copy of this area typically have DiGeorge or velocardiofacial syndrome. There's other names for it as well. This is a little girl that has that. You can see an abnormal ear here, cup-like, and a very long nose. And those are very typical of deletion of 22q11. But there are many genes, so there's like 50 genes in that area, but these two genes are the ones that are most important. The TBX1, if you have a deletion there, then you get the cardiac, the heart problems, and other birth defects. And the COMP gene is when you get the behavioral problems. People with deletion of COMP can have anything from seizure disorders to intellectual disability, autism, schizophrenia, many other brain disorders. And again, the exact identity of the genes in the segment is what's important, because it's not what chromosome it's on, but which genes are affected that really make the difference. About a year ago, I was evaluating with the help of a physician who was on the other side of the country, told me about a 19-year-old that had autism but was doing fairly well with autism and was in special schools and was involved with family and doing okay, and then decided a couple of years ago that he didn't really want to speak. In the last year, he's hardly spoken at all to anybody. Every single doctor said, well, that's autism. But a lot of you are saying, well, no, that's not really autism. There's something else going on there. Previous testing had shown that he had a deletion at 22q11, and he did have some birth defects, but a normal heart related to that. And everyone thought that this was the cause of some of the birth defects, and that the autism was the cause of the behavioral changes, and there was nothing else going on. But Actually, deletion of this area is a high risk factor for schizophrenia, and about as many as 1 out of 3 people with this deletion will develop schizophrenia and related problems. A teenager who suddenly decides that they don't want to talk to anybody is at high risk for schizophrenia. And based upon the genetic information, we were able to provide the appropriate diagnosis and to get them to the right expert for treatment. Angelman syndrome is based upon a deletion of chromosome Q11.2, and it is based upon an abnormality of this particular gene, which is a gene that's involved in protein turnover or getting rid of old proteins so that you can bring in new proteins. These are some of the issues. So you can get mental retardation and autism, absent speech, epilepsy, ataxia, which is like a baby would walk or a drunk person. But you can also have bizarre laughing and sometimes hypopigmentation. You see this boy is very light-skinned, more than the parents. And this requires sometimes special testing to find. Okay, so 2 more large mutations. There's a 22-year-old male with many neurodevelopmental problems: autism, intellectual disability, epilepsy, anxiety. These all really go together, hypotonia or floppiness. These are often brain disorders which are found together. The genetics, whole genome sequencing, found a deletion de novo that's not in either parent. Of 2/3 of this gene here. This gene encodes for a protein which is involved in clustering the inhibitory receptors together in the same place on the membrane. So GABA and glycine receptors are inhibitory receptors to try to calm down the nervous system. And colibistin is a protein which kind of ropes them together so they're all in the same part of the membrane so they can act together to turn off that neuron. And these are the problems you get. Hyperkyplexia is where you have an exaggerated startle reflex. You come up behind somebody, you go boo, and then they go right like that. That's hyperkyplexia. And then, of course, this is not something that anybody asked before, but then we discussed the genetic findings with the family. They said, oh yeah, he has extremely severe startle reflex. And so we were able to make a diagnosis. We tried to give additional glycine and GABA in terms of dietary supplements to increase those. But the patient did not get better. So sometimes we come up with a finding and we try something new and it doesn't work. This is the opposite. This is a 16-year-old male, again, severe autism, intellectual disability, GI symptoms, migraine, anxiety. Migraine's a little different here. I haven't seen that in the last few cases. He has mitochondrial dysfunction based upon sophisticated testing, which you can see in over half of cases, maybe as many as 80%. Trio, that means sequencing the parents and the 16-year-old patient, whole genome sequencing, the report was negative. But the report is based upon what people know, not what people don't know. And there was a deletion of 84,000 base pairs on chromosome 8p. That's de novo. It was not in the parents, but it was in the patient. There are 2 enzymes in here. Remember on Angelman syndrome, I said it's involved in protein turnover? Protein turnover is very common to cause neurodevelopmental disorders, including autism. And the deletion, de novo deletion that wasn't in the parents of 2 protein turnover genes strongly suggests a new disorder. We're now working on this to try to prove a new disorder and to figure out more about it. But based upon this, we did try some therapy. The other thing is that the patient has a migraine and the entire family has migraine. And we also found that there were mitochondrial DNA abnormalities that were associated with migraine that were in the patient and in the mother, which described the migraine in the patient and the mother, not from the main mutation, but from other findings. We did, based upon these, we put the patient on mitochondrial cocktail and migraine targeted supplements. The idea of the mitochondrial cocktail was not just for the migraine mutation, but also to try to stabilize the cells that are having problems with protein turnover. And the mother reported significant improvements, better transitions, sleeping better, using new words, trying new foods and activities. She ran out of the mitochondrial cocktail and the behavior worsened. She tried again, and the patient got better again. So again, that this is sometimes that you can find an abnormality that you can then figure out what to do and it works, and sometimes it doesn't work. In my experience, about half the time that we see a patient and do the genetics that we're able to help the patient is better. Better doesn't mean all better. I mean, it doesn't mean that they're cured. They still have autism in most cases. But in some cases, the autism is better enough that it's better classified as ADHD. But in general, about half the patients improve based upon this method. This is from Scientific American, that there's common mutations account for the bulk of autism risk. So far, what I've been showing you is I've been showing mutations that are so severe that they actually cause the disease and that they're rare. But there's also common mutations, common sequence variants that can be inherited from both parents generally, that each of them increase the risk. The patient inherits some from the mother, some from the father, and inherits a risk. And this is actually more than 50% of autism appears to be based not on one gene, but on multiple genes, what we call polygenic. So I showed you this before. I told you, small versus large. Common is present in a lot of people. Over here is rare. On the very far end of rare, it's never been seen before. Sequence variants that's in the patient. If it's de novo, it's not even in the parents. If the variant causes the disease, if it's monogenic, the variant by itself without any other genes is the cause of disease. Obviously, that's got to be a very rare variant. If it's common in the population, it's not going to cause autism. But if it's common in the population, it can be a risk factor. So one of the things that we look at variants is if they're very rare, they could be normal variants that are very rare. Everyone has normal variants that are rare. They could be variants that are risk factors, but they could also be variants that cause disease. When you're looking at the more common variants, you're looking at risk factors with lesser risk. It's not like most people have it, but only some people have it. In particular, with families in which both parents don't have autism, probably in many of those cases that there is a risk factors that were inherited from both parents. Of course, in other cases, it can be a new de novo mutation, which the parents don't have. So here's a more complicated autism case. Zach here does have autism. He presented at 1.5 years of age with developmental regression. He lost his language skills. But it wasn't associated with anything that we can identify that happened at that time. He also has cyclic vomiting, in which he had nausea and vomiting that would last for several days. He would be hospitalized, so then every month it would come back again. He had several hospitalizations for bowel cleanouts, and he had rhabdomyolysis where the muscle would break down. But the major issue when he came to saw me was complex regional pain syndrome. He was in a wheelchair and on high doses of narcotics and extreme pain in one of his legs. And nobody could find out what was going on. We put him on the mitochondrial cocktail and amitriptyline, which is a drug that helps with neuropathic pain and some other things. And his breakdown of muscle, his cyclic vomiting, his pain syndrome all went away. He was having more than 25 admissions a year to a hospital, and they went down to 2 per year for bowel cleanouts. So mostly on the mitochondrial cocktail, but also on amitriptyline, he went from being completely disabled with a purple, painful leg despite on 3 different narcotics, to being off of narcotics, no pain drugs whatsoever, and able to walk and run. Later, we did genetic testing on him and found that he had a change in the gene that makes acetylcholine. Acetylcholine is one of the important neurotransmitters. I believe that this was probably related to Zach, because Zach had episodes in which he would be like completely out and that's common with acetylcholine. He also had very severe POTS, or problems with the autonomic nervous system, and that's also a problem with acetylcholine. And based upon that, we use drugs that keep acetylcholine in the synapse longer. I can't make his brain make more acetylcholine, but what I can do is I can stop it from being broken down as quickly. And then he improved dramatically. Before he was put on this drug, he would have echolalia. If you said something, he would say it back like a parrot, exactly what you said, but no other language. On this drug, he was able to speak in sentences. I know that sounds bizarre. It doesn't always happen. I've known this mom for many years. I had to see it myself to believe it. It really made that much of a difference. Okay, I'm an animal lover, and these are kittens that were born at my house because during the quarantine of the pandemic, a pregnant mom cat showed up at our doorstep, just about to deliver. And what I wanted to show is not only are they very cute kittens, but at first glance, it looks like an 8-eyed monster. It's hard to see where one kitten ends and the next kitten begins because they like to cluster together. And this is really what happens in autism as well. You do get the monogenic cases, but most cases are more complicated. One I want to show you is Dane here. He was normal in the first 5 months, and then he started having episodes of regression. Not one episode of regression like the other cases, but he had 10 to 15 episodes of regression. You can see that he had, quite severe problems. He had some GI issues as well, seizure-like activities, and fatigue, constipation. And the mother has migraine, chronic pain, and GI issues. So every time that he would have a regression, he would lose skills, and he would lose whatever he learned. And then he could learn some things, and then he'd have another regression, and he'd be back to where he was before or even further down. So you can see he went from a pretty normal baby that was smiling and looking at people to be completely out of it. Exome sequencing, looking at all of the genes, found a de novo, again, not in the parent, in a GABA receptor gene. GABA is an important inhibitory neurotransmitter. So an abnormality in a GABA gene means that you have too much excitation. And that fits with epilepsy and anxiety and autism. And so that is the cause of his major disease, a defect in the GABA receptor. But we also found a variant that came from the mother in a calcium channel variant. And this gene is common in weird migraine type things. It also can cause periodic paralysis. And Dane had periodic paralysis. And it appears that what's happening is that this GABA receptor gene is the main reason why he has autism and neurodevelopmental disorders. But his deterioration, his regressions, are due to the calcium channel. And we were able to put him on treatment of the calcium channel, and he stopped regressing. He still has autism, he still has intellectual disability, and we're working on that to try to find a treatment for the GABA receptor. But the treatment of the calcium channelopathy did stop his regressive episodes. It's just to show that sometimes you have to piece out the different kittens from each other to figure out, and if you look at each kitten individually, it kind of makes sense, but all together doesn't make that much sense. That if you find one mutation, you can't just stop there, because most people have variants in many different genes which are related to the disease. And this is not just for autism, this is not just for brain disorders. If you look at diabetes, asthma, heart disease, any of these common problems, it's generally not one mutation in one gene, but multiple variants in multiple genes. And so you really need to sequence everything and tease it out. So when I talk about sequencing, sequencing started at looking just for the mutations themselves that are known. Like in Fragile X syndrome, which can cause autism, you look at for one particular part of one gene. Then around the turn of the century, we were doing single gene sequencing. There are 23,000 genes. At that time, it cost about $5,000 to sequence one gene. And that was a big money loser for the labs because it just took so much time to do that. Then as it got cheaper, we started to put panels together, maybe 3, maybe even as much as 10 genes that cause the same sort of thing. Like there's about 10 genes that cause Rett syndrome. And so you can put those genes into a small panel. But then for autism, you needed a big panel. And those panels were, you know, they may have started off at 30 genes, but soon panels were hundreds of genes or even 1,000 genes for autism alone. So then things, as things got cheaper with the sequencing, we went to whole exome sequencing, which is sometimes called exome or WES, W-E-S for whole exome sequencing. So this sequences the protein coding segments of all 23,000 genes. So you can read what the sequence of all of the genes would be. And in academic circles, in university hospitals, this is what they'll often will do with cases they don't know how to handle. Now, 98% of the genetics is not in the exome. 98% of the genes does not code for proteins. Whole genome sequencing, genome or W, whole genome sequencing here, looks at sequencing all of the DNA, including the 98% that doesn't make for proteins. Now, when we first started that, almost all of the diagnoses were in this area here, the exome. But now, as more and more whole genome sequencing is done, we realize that a lot of mutations that cause disease are outside of the exome. It may only be maybe 20, 30% or so of the mutations that are known are outside the exome, but it's not a small number and it's increasing dramatically. In particular, like the pharmacogenomic variants, the variants that say that this drug will be helpful in the patient likely and which dose to use, those tend to be outside of the exome. So I not only use sequencing to figure out if I should use a medication, but what medication I should use and what dose I should use. Because there are proteins which are involved in making enzymes that break down drugs. And it's important to know if you're a hypermetabolizer, you get rid of the drug really fast, so you need a higher dose. Or if you're a hypo, a lower metabolizer, you break it down very slowly, and you need a low dose. I've seen many patients that tried a drug before and they said it didn't work, but they were fast metabolizers and they didn't use a high dose. A high dose worked fine. And I've seen many other patients that say, oh, I don't want to try that again. I had horrible side effects, but they were a slow metabolizer and a normal dose was toxic. You can just use a quarter of the dose and it could be very helpful. So there's a lot of things that you get from whole genome. This is what I recommend. Is also if you do whole genome, remember I showed you it gets large and small. So you get the small sequences, you get the large sequences, and you get mitochondrial DNA as well. If you order this, you have to order all 3 of these tests separately. It's also, you can get Fragile X from whole genome because genome sequences everything. So when you actually look at it, yes, genome is more expensive than exome, but genome is less expensive than doing all the tests that you need to do independently. And this just goes, see, this is in alphabetical order here. This just goes to show that the type of computer readouts that I look at, this is for, I can click at different things and get more information, but each variant in the gene, and this is an Excel database, and that has about 40,000 lines to it. In this slide right here, I took all of the variants that I thought might be of interest or importance. This is Dr. Temple Grandin and her genome. So all of the variants that I thought might be important in Dr. Grandin are on here. And she has a mutation, her major mutation. She's a very famous person in the United States with autism. She lectures around the world. She has a mutation, where did I find it here? It's on here. No, I can't find it right now. It's because she has a Shank mutation. It starts with an S, so it's off the slide. That's one of the major reasons why she has autism. But you notice she also has mutations in the familial Mediterranean fever gene. And these variants here are actually what caused her most of her grief, which caused a lot of her symptoms. So genetic counseling, when we talk to people about genetic testing, we want to know, why we're testing, what tests we order, what are the alternatives to testing, samples. The alternatives, there's really no alternatives to testing right now. You can test less, you can test not at all. Samples historically was with blood, but I almost always use saliva now. Kids would much rather spit into a kit than they would to have their blood drawn. Kids that can't spit because their autism is very severe, you can use a swab on their cheek. What does a positive result mean? It might mean that we find the result. It might mean that we have treatment. It might not mean any of those. What does a negative result mean? It doesn't mean you don't have autism. It doesn't mean it's not genetic. It just means that we're not clever enough of the 40,000 variants that we find in each person to figure out what's going on. What does it mean for relatives? Obviously, if you have a genetic abnormality, relatives might have it. There may be some implications for that. Limitations of testing, like you can't find everything, and we certainly don't understand everything. There's also like the costs, reporting options. There's many other things, but these are the sort of things that are important. When we're talking about off-target or incidental, if you're testing a child because of autism, if you find something that relates to heart disease or cancer or a bleeding disorder, those are incidental. That's not why you did the test. Those are considered off-target. I don't find a lot of problem with that whatsoever. In fact, in 30 years of doing genetics, nobody ever told me with my patient they wish they didn't know something. The vast majority of my families want to know everything, and they're happy to get incidental findings. Often they're helpful. And the common type of incidental findings that we see are like if you try this drug that you need a lower dose, or this drug won't work, or this drug may work very well. And occasionally we find something that says, if you try this drug, you likely will die. That happens. With Dr. Grandin, she has a mutation that if she's given warfarin, which is a very common blood thinner, Coumadin, that she will likely die. She'll just bleed out. She calls it like a stuck rat, like stuck pig, when she lectures. So she knows never to go on that drug, that she probably will die if she does. So these are the kind of things that you can get for incidental findings. Sometimes we find bleeding disorders that if you go to surgery, you need an antidote first. They're far more helpful than they are a problem, and they do occasionally find them. I would say a significant incidental finding probably every few patients or so, but usually I don't find I'm not looking for things I can't do anything about. There's 40,000 different variants. When I say something to families, it's usually because this looks credible information and there's something you can do about it. Consanguinity. All of us are related to everybody else on the planet by 180,000 years ago. When I look at the genes, I generally find that the parents are related to each other more often than not, if you go way, way back. But that's not what I'm talking about. Sometimes I find that people are cousins, and occasionally, but very rarely, we find that they're even closer, but that would be abuse situation. But every once in a while, I'll say, well, you know, the parents are third cousins or something, and they didn't know that. That's not uncommon at all. Misattributed paternity. Daddy is not really daddy. That happens sometimes. So I haven't seen that in about 5 years or so, but I used to see that before people realized how powerful DNA was. Mothers that were at risk would provide DNA testing. I haven't seen this in a while. And disclosure, what are your options in terms of what kind of findings that you want? And we kind of discussed that a little bit before. Here's my daughter last year. And this just shows you that even the desert in California can be really beautiful sometimes. This is the super bloom from all the rains that we had a year ago. So how can your child take advantage of the genetic testing that we've been talking about? Well, prices have dropped. I actually calculated that prices have dropped 1 million-fold. 20 years ago, it cost 1 million times more money to sequence each nucleotide. This is the price that was insurance companies and that families would pay. It's because we would sequence one gene and it would cost $5,000. Now we sequence all of the genes and it costs even less, a little less than 5,000, sometimes as low as 1,000. It's actually 1 million times cheaper than it was 20 years ago. And even than 5 years ago, it's 1,000 times cheaper. The price, you'd say, well, it's going to be free if you wait another day. It's pretty much hit bottom. I see that prices this year are almost exactly the same as they were last year. because it's not likely to go down anymore, because the sequencing itself is not the cost. The cost is the interpretation, the cost is the marketing, the cost is the personnel involved. It's not the sequencer itself anymore. The actual sequencing is so cheap now that the other costs are the vast majority of what you're paying for. So I know I have seen companies like Dante in which you can get whole genome sequencing for as low as $300. We are still working on the technology to try to take that information and to translate that into something that I can read. The IT people are having difficulty at that. I hope to get there. Right now, the cheapest exome that I know about is Centogene in Germany, and I send patients there. I don't send them to Germany, I send their saliva to Germany to be sequenced. And then of course the results are on the internet. You can get, for self-pay, you can get exome in Germany for as low as $1,000. And that is a quality that the IT people can put it together. I can look at it and it's a good exome. Again, that's not genome, this is exome. We can do mitochondrial DNA and CMA, chromosomal microarray, for as low as $500 each at Centogene. And so that's 2,000 together. You can do genome for 2,800. That's variant YX, which sequences in the United States and in Israel. For India, there would be in Israel if they do the sequencing. So it's not much more expensive. And if you add in the pharmacogenomics and the Fragile X and other things that you get that aren't in here, it's actually cheaper to do whole genome sequencing. Now, if you're going to do trios and you're going to do the parents, it's generally about double the cost of doing a patient's cells. And then there's interpretation costs, of course, of the doctor that's going to read that and talk about it. In the United States, most laboratories will take insurance. I don't know how the insurance market works in India in terms of covering these sort of tests. But interpretation is the most difficult step. A machine can put the entire sequence of the genome onto the internet in an hour, but you have terabytes of information, so much information that you'd have to mail a computer hard drive to send that information. You can't even, that's the only way to do that. It's finding somebody that can interpret that information and to make diagnoses out of it. That's the hard step. So I do recommend whole genome because it covers all of this, and all of this is important. We've discussed each and every one of these tests before. So the next question is a singleton. That's just doing the patient versus a trio. So singleton is cheaper. It's generally about double the price to do the parents as well. It's not triple the price because, again, the interpretation is the difficult part, and we're not interpreting the parent's genetics except to see if the child sequences, if the parent has it. If a parent does not have the sequence, then it's an apparent de novo, a new mutation, and those often cause disease, as you've seen several of them. So there's many advantages of trio testing. The main one is to find de novo variants, or variants that are not in the parents, a new mutation which causes the disease. If it's a recessive condition, we want to make sure that both variants came from each parent, that both parents are carriers with one each. If a patient has two variants in a gene, but a parent has two variants, then that's not going to cause the recessive condition. And of course, if other people in the family have problems, it's like a bunch of people have migraine, it's a good, if you have the parents, you can see, well, at least it comes from the parent that you think it should, and then you can trace it through the family. So I do recommend trio for any except for the most basic cases. If it's just learning disabilities or ADHD, you can probably do singleton, and I do that sometimes. But I do recommend trio for any except for the really basic cases. If you have epilepsy, severe autism, intellectual disability, birth defects, or dysmorphic facies, you really need trio because new mutations are pretty common. So again, I mean, only your physician can determine which test is right for you. However, in most cases with autism, trio whole genome sequencing is the correct test in my opinion. I do work with physicians. They tell me about patients and I suggest testing. Sometimes they say that there are financial concerns. We can't do everything. What would you recommend? And so one of the things that our center does is to work with the family and the physician to give a little bit of information and to say what kind of testing. We also can facilitate, well, how do you reach the laboratory? What do you do to sign up and everything? But then again, who will interpret the test is the key option, is a key issue. This is one thing that we're doing throughout the world. We've done it in several countries so far, a peer-to-peer counseling system. I don't have a medical license outside of the United States. In fact, I can only practice in half the states of the US right now. Outside of the US, I cannot talk to families. I cannot see patients. But I can talk to doctors and help them order the right tests. I can talk to them and to interpret the test, and I can help them to try to see how to treat what we find together. And so that's what the peer-to-peer counseling service is to set up a communication between us, Neurogenomics at NeurAbilities, and your physician, and to set up so that I can help them, we can help them order the test, and we can help them interpret the test, and also to go over what the treatments there are. So it usually is, we're on a telemedicine call like this, and then we talk about the family and we talk about the patient, and I usually have some medical records that I looked at ahead of time. So the doctor tells me about the patient, and then I tell him or her what I found in the DNA, and we connect the two. The DNA it won't help without the patient doesn't help without the DNA. It's the connecting of the DNA and the patient together. The doctor, your doctor and me together are much better able to figure out what's going on than either of us alone. If you're interested in this service, here's a phone call. I put the one here for the United States, fax, and then here's the email, which is the most important one to know. We've done about 100 cases so far, and we're actually putting the data together because we want to publish it. We did put an abstract and a meeting of the first 17 patients that we did. Of the first 17 patients that we did this service on, 94% the physician changed what they were doing based upon what we discussed. 52%, half of them got a diagnosis. This is what your child has. And there were recommendations that change in therapy. Of the 17 patients, 15. Now, when we put together the next 100, these numbers are pretty similar. In about half the patients, you get a diagnosis, and you change therapy in most patients. This is to indicate here, outside consultation. A lot of people are afraid, we're going to find all these variants of uncertain significance, and you're going to have to do all of these things. That actually didn't happen very much. By far, the most common reason to do additional testing was the patient was sequenced, but not the parents. That was most of this. Some of this was to do like an EKG or an EEG or something, but most of it, nothing, we didn't suggest anything invasive, no surgeries or anything. And outside consultation was 23%. Two of these were cardiologists because of variants that might affect the heart. And then I don't remember what the other two were, but it's not like we're going to find 100 different variants and 20 of them you need to follow up on. That just doesn't really happen. This is from ancient Greece, the fates. In ancient Greek mythology is that there's nothing that you can do about it. The hero is doomed to die because the fates have determined the length of the life. Your genes, you can't change. Reading your child's genes can be scary because that's the destiny. You can't change that. But it matters how you play the game. Think about a card game. A good card player can win with a bad hand. A bad card player can lose with great cards. The genes are there. It's like the cards you've been dealt. You can't change that, but you can play your hand better. You can play your hand worse. Genetic testing tells you what's there, but it doesn't tell you what's going to happen because you can play your hand much better. Getting testing helps you to try to get your best potential. It doesn't tell you there's nothing you can do. How does testing make a difference? Well, I've already said the right therapy might not be a drug. The right therapy might be cyclocreatine. It might be alpha-ketoglutarate. It may be something else. It may be diet. Right drug. Not just throw drugs at the problem, but find the drug that's actually going to help. Sometimes the right drug is something that the physician would never have thought of. I never would have thought of Kytril. And the right dose. Is it a smaller dose? Is it a larger dose? Is the right amount? All of this can be helped in many cases by DNA testing. So this is just to show you that, new variants that are not in either parent are pretty common. About 1/3 of autism patients have it. They are more common in the lower function individuals, but you can find it across the board. Inherited variants, you can be from a completely normal parent, or a parent that shows very mild issues, or somebody that has more significance. And I wanted to show you this case report is because mitochondrial abnormalities are so incredibly common in autism. And that this girl, in fact, you've seen her artwork throughout this. This is Kelly. And she has autism, but her main problem was migraine. She had migraine 24 hours a day, 7 days a week, 365 days a year. A lot of other issues too, fatigue, bowel problems. You can see this in a lot of the patients. Biochemical testing revealed the signs of a mitochondrial disorder, and she got much better on a mitochondrial cocktail. Later, when we did the genetics, we found 2 mutations in the gene here, this enzyme, and that makes alpha-ketoglutarate into the Krebs cycle. We gave her alpha-ketoglutarate, and this is the email that her mother sent, that she improved quite a bit on alpha-ketoglutarate. And again, alpha-ketoglutarate might be the right treatment, and that's part of the mitochondrial cocktail, but not one of the supplements that she was on at the time. So most patients with autism have mitochondrial abnormalities, and I think that I've shown you before, several of them before. So, creatine disorders, in Pam, and in Carter had a TRAP1 abnormality that's involved in a mitochondrial chaperone. Those are mitochondrial disease that was proven with DNA techniques. Same as with Kelly I just showed. But even most of the other patients, they got better on a mitochondrial cocktail. And my patients was getting tired of telling them to get like 10 different things. Some of my patients were on as many as 40 different supplements. Because to make the mitochondria work better, you have to give a lot of different things. And so I came up with a formula here I called Spectrum Needs. It promotes mitochondrial metabolism. It has 20 different components to it. It also is a very high dose multivitamin in general. It includes creatine and alpha-ketoglutarate and carnitine and CoQ and all of those things that we've spoken about, or at least most of them. Comes in 2 flavors. These are the ingredients here. But I also want to talk about CoQ. This is my own brand of CoQ, but in India you have other brands. This is ubiquinol. Ubiquinol is the important one. There's 2 different types of CoQ. Ubiquinol is 5 times more bioavailable than ubiquinone. Ubiquinone's the other one here. And these are considerations from which brand you buy. Some people don't want soy. Some people want to have, they need liquid because their kids can't swallow soft gels, the size of the gel, So these are considerations. But I always recommend, in addition to a product such as Spectrum Needs, to use ubiquinol on top of that. And a lot of patients improved. In fact, I've been using mitochondrial cocktail way before I had my own product 2 years ago. I was using it in autism for 25 years. And a lot of the patients that you see that I've shown you said they got better on mitochondrial cocktail, and then we did the genetics. Well, it's because they got better on mitochondrial cocktail 10, 15 years ago. And then the genetics became available. And so then I found the exact problem. Sometimes it's in the mitochondria, sometimes it's not. But mitochondrial dysfunction is as much as 80% of autism. And it's one thing that you can do while testing is getting ready. If you can't do testing for whatever reason, it's one thing that you can do. So here is the last slide. This is where you can get information about what kind of services that we can offer. It's our website here. There is a published article on genetic testing in autism. It's written for physicians. I actually just this week wrote another article for families that we can share later when it's edited. But this is that you can just take this direct link and take a look at it. I believe that these slides are all going to be available, so later you don't have to like write down all those numbers really quickly. And that if you're interested in neuro needs, and I am the medical and scientific director, so that there is a conflict of interest of my own formula. But anyway, this is how you reach NeuroNeeds. Just www.neuroneeds.com. So I will leave it on here and then open it up for questions.
Thank you. Thank you so much, Dr. Boles of NeurAbilities Healthcare. Dr. Boles, we have a lot of questions. I don't know that we're going to get to all of the questions today. But as you said, this presentation will be made available through both the India Autism Center and NeurAbilities Healthcare on our YouTube channels in about a month. I think the #1 question that we're getting is, what is the best age to do genetic testing?
Now. There's no reason to wait. Every year, genetic testing got better. And if you waited another year, then it's like there would be a better test. But now we do whole genome. Genome is everything. It's not like next year we're going to do more. There's nothing more to test. We'll understand more, but the sequence will be on the computer, and then they can take another look at it. There's no reason to wait. The genetics was the same genes were there since conception. Any time of your life. Any age. Any age. We do babies, we do adults, everything in between.
And I think that another point to clarify, people are asking, what are the costs if they're doing this testing in India, and how do they access a lab? But I think that what you said is they can work through their doctors in India to collaborate with you if they are interested.
DNA testing in the vast majority of the world needs to be ordered by a physician. So your physician has to order the test. We can help that physician determine what test and where.
Okay. And if they want more information, they can email us at info@neurabilities.com, and we'll be sure to get that email and that question to you and to your coordinator. Here's another question. Do brain scans help provide any additional data along with genetic testing?
Brain scans. We used to do, not that long ago, it's a genetic approach in which you would get all the information about a patient, and then you would say, okay, these are the genes we want to test, and they would test those genes. Now we use a genomic approach, which is to test all of the genes, and then we say, okay, these are the things that we saw in the genetics. Now let's go back and do the tests and to see what's going on. So in the past, let's say we found a migrational defect in which the neurons didn't migrate to the right place, then we would sequence the genes that cause migrational defects, or whatever we knew at the time. Now what we'll do is we'll do the sequencing. We say, it looks like that there's a change that may be disease causing in a migrational gene. That now let's go get the MRI or the scan of the brain to see if there is a migrational defect. So sometimes the scans are helpful to understand the genetics. The scans are, I mean, if you have to sedate the child, there is a risk. And the scans are expensive, and the scans can be as expensive as the genetic testing. I'm not a neurologist. I don't use the scans to a large degree. Sometimes they can be helpful, but I usually start with the genetic sequencing and then go from there.
Do you recommend for all children with neurodevelopmental disorders?
Recommend what for testing, you said?
Genetic testing.
Yes. In patients with the very, very mild end of the spectrum, let's say they have difficulty reading, but they're otherwise completely normal, or they have mild ADHD, sometimes we can find a treatable abnormality, a lot of times we can't. So those would be ones that I would say would be optional. It's not to say it's not useful, but just to realize that you're not going to get the 75% chance of finding something that or 50% that we've been talking about. But I do recommend it across the spectrum.
Okay. Let's see, so a couple questions left. Would medication, if medication is prescribed, is that something that's typically needed to be continued through your lifetime, lifelong?
There's thousands of different medications and it's very complex. I wouldn't stop a medication that your physician thinks that you should be on until discussing that very carefully with your physician, or at least getting a second opinion if you don't agree. If a medication is causing side effects and it's not helping, then it's a discussion you really need to have with your physician, should you continue. If you've been on the medication for years and you don't know if it's helping or not, have that conversation. Sometimes people will stop a medication and the patient will get worse and they say, oh, that's why I need to go back on it. They go back on it and the child's okay again. So it's a difficult situation, but I would say is that anytime you're on any drug whatsoever, you should have that conversation at least every other year, if not more frequently, with your physician as to, do we still need this?
Can you comment, if one child has autism, is there a way to predict the probability of a second child being diagnosed with autism?
Without genetic testing, the risk is about 25% in a boy and about 6% in a girl. So if everybody in this audience, let's say everyone we're talking about who has a child with autism, another child would be about 25%. Now, some of the families will be almost 0%. Well, it won't be zero because there's always the chance of a new mutation or something. And some of the families will be quite higher. But overall, it's about 25 in a boy and 6 in a girl. With genetic testing, if we find a de novo mutation, it goes to less than 1%. If we find a recessive mutation, it's exactly 25%. If we find risk factors in both parents, it might be higher than 25%. So it really, genetic testing can make the risk higher or lower. In particular, it can make it lower if it's a de novo or a new mutation. But these risk factors that I told you about, the 25%, that's for autism. If you look at all neurodevelopmental disorders, it's as high as 50% in boys. So they're about 25% for autism, about another 25% for something like a learning disability or ADHD or something like that. So the risks are high. And genetic testing can sometimes change those risks if you find the problem.
Okay. This is probably a difficult one to answer, but I'm going to put it out there. Any chance of a cure?
A cure means is that the disease is gone, like you give an antibiotic and the bacteria is dead. At this point, cure would be no, but I do have families who are on treatment whose kids don't have autism anymore. Carter that I showed you, the first patient, he doesn't have autism anymore. He has some mild learning disabilities and he has ADHD, and he's treated on ADHD medications. I didn't cure his autism. I found the cause of his autism, put him on a treatment, and his autism is better to the point he now has ADHD. His disease is not gone, but it's better. It's more than 50% better. Certainly not everyone has that degree of improvement. When he stops, and like I said, 5 times he stopped the treatment, he went back to where he was. I don't see a cure, but finding the exact cause and treating, we're already there with some patients.
Okay. It is way past time. I think that this is a great place to stop. Want to thank you so much for your time, Dr. Boles.
Oh, you're welcome.
Thank you. And I also want to thank the participants for tuning in to the Autism Specialty Care Education Series brought to you by the India Autism Center and NeurAbilities Healthcare. If you would like an e-certificate or would like to leave feedback for us, please fill out the Google form attached and we will send you your certificate within 4 working days. Please note that the form link is only valid for 24 hours, so make sure you fill it out within that given time frame. And in case you were unable to fill out the form, please contact us on the WhatsApp on the number given on the chat slide, or email us at info@indiaautismcenter.org. If you would like to stay updated with other webinars, please follow our social media handles or follow the link in the WhatsApp group. The recording of this webinar will be available on our YouTube pages within 1 month from the date of this webinar. And we look forward to your suggestions and hope to see you for our next 2 webinars. And we'll stay back a few minutes to answer any questions you may have, and you can use the chat box to address them.
Thank you very much.
Thanks, Dr. Boles.
Dr. Boles.